All right. Here we go. Take two. I've got two cameras going on here. Not sure which one's going to work, so I don't know which one will work. I might be alternating between the two, so don't be freaked out. On today, and whatever day you're listening to this, on Friday, we went through this was where we ended with every class. The base pairing rules to how DNA is going to match up with itself. The rules that adenine will always match with thymine, and guanine will always match with cytosine. In a few classes, we actually went through why purines and pyrimidines must always match. That's the second rule of base pairing. For those classes that didn't get to it, I will explain, but I do need some student volunteers within the group. For today's lesson, what I want to teach is the idea of a more detailed structure of DNA and why it's structured the way it is. So this box you have in your notes, I think it's actually easier if you are going to flip it towards up and down, because I'm going to draw it sideways. It's just more conducive to draw it up and down, but I had the box sideways because it's just easier. So then let's go to, I don't have the box. Imagine that I'm drawing this within a box that is more, now your paper is flipped. The first thing we're going to do is we're going to draw and redraw a nucleotide. So remember the parts of the nucleotide. What we have, we're going to start on the left-hand side, so put this more towards the upper left-hand side of your box when it's upright. Draw the phosphate group connected to, remember the phosphate group connects to the 5th carbon on that 5-carbon sugar. Off of the 1st carbon is where we attach the nitrogen base. For the first one, we get to kind of have the creative luxury to draw them. Because I'm drawing 2 rings, it limits it to it's a purine. So that can either be the adenine or guanine. So this is one nucleotide in a series of the left side DNA strand. So then what we do is we're going to draw a second nucleotide underneath this one. So draw that as well. So another phosphate connecting to another 5-carbon sugar. And this time, let's arbitrarily make it into a pyrimidine. Alright? Nucleotide is stacked on top of another nucleotide. They are not chemically bonded, so we haven't built yet a polymer. I know when we used to be building polymers, we would talk about that whole process of dehydration synthesis. We remove a water. That is to assume that it's happening here, but we're not focused at the molecular level, so we can't technically see it. When we were doing monosaccharide to monosaccharide, we were looking at the atoms, so we were able to see where an OH was removed from one and an H was removed, so they can chemically come together to make a disaccharide. These are more simplified diagrams, so we can't see that. We have to, again, assume it's happening. So notice, this phosphate and this sugar, they're very close together, but they are not bonded together. So what we're going to do is we are going to connect that. I can kind of change the color, you don't have to. I'm going to change the color just to show you that this is a kind of new connection. That red line there is going to show that these are now chemically bonded. There's not a specific name for it. We can kind of say it's a phosphodiester because it does involve a phosphate to a sugar. But notice where the connection happened. The phosphate, now what number did it connect to? Remember the number system? 1, 2, 3. So the phosphate on the next nucleotide down connects to the 3rd carbon in that 5-carbon ring. So let's just add another for how much space I have here. I'm going to put another one. So another phosphate connects to a 5-carbon sugar. And then here, pick what one you want. Pyrimidine, purine, what do you want? And for those 50% that said, let me do a purine again, I went with you. For the pyrimidines, sorry I couldn't read your mind. Let's connect once again this newly added nucleotide, connected 3rd carbon to the phosphate. So now I'm building a polymer. I have 3 individual nucleotides, all chemically bonded. This occurred through dehydration synthesis, this occurred through dehydration synthesis. So what you can do is you can draw 3, you can actually draw more if you have more space. I'm sure many of you will just stop at the 3rd one because that's where I stopped. The key here is now we're going to identify something called DNA has 2 sides. They have a side that is called the 3' side and a side that is called the 5' side. And it's all based on those numbering of the carbon. So if you go back up to this one and I see this is the 1st, 2nd, 3rd, 4th, and 5th. It's very easy to identify the side that you're working with. It's the number that is closest to the end of the molecule. So here, this is the end. This is the end of it. So what number is closest to that line is the 5th carbon. So what they say is, this is the 5 prime, and I can explain prime, it's just a number that they associate with these carbons. They call it the 5 prime side. You can say it's the 5 side, you would still be able to answer any question that deals with it. So this is the 5 prime side because the 5 is closest to the end. If you go down to the other end of the DNA molecule, ours is a very short one, it's only got 3 nucleotides, and we number the carbons associating down here, 1, 2, 3, 4, and 5. Notice, of the 2 numbers, we have a 2 and a 3 closest to this line. They will go with the 3, and they will call this the 3 prime. That little apostrophe there is what they designate as the prime. So this DNA strand has a 5 prime side and a 3 prime side. What we'll now do is we are going to build the other side of our DNA molecule. But here's the issue. If I drew a nucleotide, I don't want you to draw this right now. If I drew a nucleotide in the same fashion with a 5 prime on this side closest to the end, remember the nitrogen base has to come off the 1st carbon. Here's the thing. What bonds one side of a DNA to another side of a DNA is the nitrogen bases. Notice the nitrogen bases are on opposite sides. They're not even going to want to connect. So what we have to do, and hopefully again you did not draw this, we're going to erase it. Instead, I have to flip around the right side of my DNA so that my nitrogen bases will connect with one another. I'm going to show you what that would look like. I have to do this upside down. You can actually flip your paper upside down and it will actually probably be drawn a little bit easier. So I'm going to first draw my sugar. Notice now instead of it being pointed at the top, I'm going to draw the base as being more flattened. Because the point to my 5 carbon sugar is going to be a little larger. There's the point. It's kind of flipped around. It's going the opposite direction. My numbers have to stay consistent. So if this is the 5, because that's where the point is, in the clockwise motion, this is 5. So here's my 1st carbon. It's on, so 1, 2, 3, 4, 5. It's probably even easier to see when you actually flip this upside down. So there's my, I'm sorry, it's really a lot bigger than the other one. How about I erase that quickly? I guess that's the part where I would have said take 2, but I don't have the patience to do that. So this is my 1st carbon. Recall that off of my 1st carbon is where I have my nitrogen base. So 1, 2, 3, 4, 5. Off of my 1st carbon, this is a purine, so this has to be a pyrimidine. Now the nitrogen bases are connecting, they're facing one another, they're allowed to bond. So then off of the 5th carbon is where I connect my phosphate group. So notice, it's an upside down nucleotide, and that's what we're going to talk about, is going to be a term related to that. So continue and draw 2 more underneath it. You can see it's a lot harder for me to draw upside down. This is a pyrimidine, so therefore this has to be a purine. There's my phosphate, and my final one . This is definitely a lot harder side to draw. Purine, this has to be a pyrimidine. Off of that, there's my phosphate group. So now I'm going to connect just like I did the other side, so I'll change it to red for my diagram. There's my phosphate. There's the 3 carbon. Remember up here? The 3rd carbon is the upper right. There it is, closest to the phosphate. Closest to the phosphate. So now I have 2 sides of my DNA. With a little practice, this could be an adenine. I'm going to make that up. I know it's a purine, so it's either A or G. If that's adenine, you should now be able to tell me what this one is. Good. Thymine. This, I'm going to say, is cytosine. So therefore, this has to be guanine. Good. And then this one, let's say this is guanine, so therefore this is cytosine. This is now a full DNA molecule in a little more detail. Nitrogen bases have to connect, and they connect via a bond that now we're going to talk about. That bond is a hydrogen bond. Remember from hydrogen bonds with water, it's a weak attraction. So there is going to be an attraction, probably between like an oxygen of one nitrogen and a hydrogen of another. Basically like a water. What will happen is, so I'm not going to draw a full line because it's more like an attraction. And there's a reason why that is. I'm drawing two of them because between any A and T, there are two hydrogen bonds. That's not a double bond, it's two different hydrogen bonds. Between G and C, you will get three of them. It's just based on the atom's arrangement that dictates it, nothing we can control. So this one is another G to C. There. You can label this is a hydrogen bond. The term that's associated with this diagram is something called antiparallel. For math, you know parallel just means that it never intersects. If these nucleotides, if I made 1,000 more of this, that would never intersect with the left-hand side. That's what makes it parallel. The term anti, a lot of people when they say antiparallel, they think kind of perpendicular. That's not what that means. Antiparallel means it's running in an opposite direction. So in class, I'm going to show you what it kind of means using students. But for this diagram, you can see this one is facing downward. The phosphates are below the sugar, and they're running in this direction. The left side, the phosphates are on top of the sugar, and they're running kind of this. Notice we're going to end this video with talking about the 5 and 3. The side that is closest, the carbon that is closest to the end is not the 5 anymore. This is a 3' side because the 3 is closest to the end of the molecule. Down here, if we read and keep the number going, 1, 2, 3, 4, and 5. So this side down here is the 5' side. That's what makes DNA antiparallel. The 5' side compared to the 3' side, they're opposite on the ends. Up here, 3', 5'. So in class, I'm going to ask you if you really understand what antiparallel means. The question as to why, it has to be antiparallel if you want nitrogen bases to actually connect. If this was parallel, if this was a 5' side and this was a 3' side, remember you've got to flip it around. So the nitrogen bases would be facing right. They wouldn't even want to connect to this. So in order for DNA to make it bond together left side to right side, the nitrogen bases have to face each other. And facing each other is only accomplished via a opposite antiparallel 3', 5' side. I hope you got that. I hope you can hear everything. And I hope this finally works or I wasted another 10 minutes of my life. Alright? I'll see you on Monday.